very good. Okay, so it's my great pleasure to introduce today's speaker at the Theoretical Biophysics, Computational Biophysics Seminar Series, Professor Reza Dastvan. He's visiting us from Doisy Center at the St. Louis University Department of Biochemistry and Molecular Biology. So by way of introduction, Reza was actually admitted to the elite program in biotechnology in my country, Iran, where he went through the ranks, BS and masters there, and then joined the PhD program in Frankfurt, Germany, where he actually switched to theoretical physical chemistry. And that's where he started applying EPR as a biophysical technique to study proteins. And then after completing his PhD there at Frankfurt, he moves to Vanderbilt University with our colleague and friend, Hassan Musharraf, and continued his work there, really doing groundbreaking work there. I remember very clearly hearing from Hassan that he completely gave up on this protein until he came to the lab and he rescued and revived everything in the lab, including P-glycoprotein and other members of transport membrane proteins. And then a few years ago, let me see when exactly. Yes, in 2019, he moves actually to St. Louis University, or SLU as you call it, where he started as an assistant professor there in the department, and just recently he got tenured very quickly actually in to associate professor, and is continuing his work there. So he's building a very nice program, one of the few labs maybe around the country who continue to work using EPR and studying protein dynamics. As you know, the probe that you use is very, very small compared to other fluorescence techniques, for example, and it doesn't affect the protein dynamics as much. So they have a very, they claim, very precise way of looking at how proteins move, which makes it actually perfect to collaborate with for us, for computational people to really describe these motions computation. Okay, I don't want to take too much time of you. Thank you very much for coming all the way from St. Louis to Urbana-Champaign, and we look forward to your talk.
Thank you. Thank you, dear Ahmad, for the kind invitation and introduction. It was a great pleasure meeting trainees here and hear about fantastic research. It's a great honor to be here. Before delving into the main topic of this presentation, I want to provide a brief overview of my research program and how where we are headed in the next couple of years. My research program is related to molecular mechanisms in metastatic cancer, and hypoxia has a critical role in the pathogenesis of cancer and major causes of mortality. And under hypoxic conditions, we have low energy status of the cell, so we have lower than normal concentration of phosphocreatine, which is one of the main cell energy reservoirs other than ATP. We have calcium mobilization, and at least we have transient intracellular acidification that could translate to extracellular acidosis. And under these conditions, certain protein kinases like AMP activated protein kinase, which is again is a cell energy sensor, they're activated, they can phosphorylate a host of downstream signaling proteins, including kinases. And my lab works on 3 of these kinases. One is brain type creatine kinase that catalyzes the reversible phosphorylation of creatine to generate phosphocreatine from ATP, and it has high abundance in neurons and metastatic cancer cells. And the other 2 are sphingosine kinase 1 and sphingosine kinase 2, which
is coming. I think your computer is probably slow.
Yeah.
Yeah.
Oh, please.
There you go.
Okay. Or otherwise I would continue there. Yeah. So, yeah, sphingosine kinase 1 and 2, that they control the cellular concentration of 2 bioactive lipids, sphingosine and sphingosine 1 phosphate, which the ratio between one of the sphingosine metabolite ceramide and sphingosine 1 phosphate determines the fate of the cell toward death or survival. So S1P, sphingosine 1 phosphate, has a key role in regulating the growth, survival, and migration of mammalian cells with profound proangiogenic effect. But the majority of its function is extracellular, so there is a dedicated family of lysolipid transporters that are called SPNS family, in particular SPNS2, that they transport sphingosine 1 phosphate out of the cell, and extracellularly, the sphingosine 1 phosphate can bind to its G protein coupled receptor and activate the downstream pro-survival mechanism. And my lab, in collaboration with Dr. Ahmad's lab, we want to combine EPR and DR spectroscopy with MD simulations to understand the functional mechanism of different members of this family, including SPNS2, as well as its bacterial homolog. And my presentation today mainly focuses on two of these bacterial homologs and our finding on that. But going back actually to these kinases that are phosphorylated under these conditions, for instance, in the case of brain type creatine kinase under hypoxic condition, they can be released from cells using an unknown mechanism, and extracellularly, they can use extracellular ATP to generate phosphocreatine, which then will be imported back to cells to compensate for the low energy status of the cell. The same is true for sphingosine kinase 1 and sphingosine kinase 2. Again, they use extracellular ATP to generate sphingosine 1 phosphate so they can boost this pro-survival mechanism. So we have another multidisciplinary project with NCI that we want to understand first what is the molecular regulation of these kinases. For instance, upon phosphorylation, we have some structures, but the molecular mechanism of the regulation is poorly studied. And also we want to understand how they are released from cells. And the hypothesis is that there might be pH and membrane curvature dependent translocation of these kinase targets to the lumen of endocytic vesicles, then they can be released. And then we are combining DIR with hydrogen deuterium exchange, high-speed atomic force microscopy, and cryo-EM to understand their membrane binding mode as well as their regulation. And while working actually on this family of lysolipid transport, we realized that some of its members, both bacterial and human, might, in addition to the specific lysolipid transport activity, might also be involved in multi-drug transport activity, especially in the case of SPNS3, which is another member of this family. It's upregulated in acute myeloid leukemia, which corresponds to poor prognosis in chemotherapeutic patients, which by working on multi-drug transport over years, actually, it seems that if our hypothesis is true, actually, we could have discovered a new family of multi-drug transporters. So we are hoping actually to start one project on this. And by working on these mechanisms, basically, we want to design modulators, inhibitors in the case of cancer to tilt this towards the death, or even in the case of, let's say, chronic hypoxia, maybe stimulators of this mechanism would be important to basically alleviate some of the chronic conditions. So my lab uses, the main technique that uses my lab is EPR spectroscopy. And for traditional EPR spectroscopy and site-directed spin labeling, we basically, those cysteines that are exposed to labeling, actually have to remove them, but we can leave the buried cysteines, or those that are, they can form a disulfide bond, and then we introduce cysteines at sites of interest, and we can label them using this label, and then the spin label is paramagnetic, so we can study that using EPR spectroscopy. But there is another way of doing it using non-canonical amino acid and using this trans-cyclooctane spin labels that basically by click chemistry, we can introduce the label without removing the endogenous cysteine, and it can be done even in cell. And Ryan Neal lab actually published a paper on it, but it's still under development. So most of the projects that we have are using the traditional way.
So
and
this
then,
you can put anywhere. This
yes,
unnatural amino acid.
yes.
Okay, I see. I see.
Yeah, and then we can basically study this label proteins, for instance, local backbone dynamics, if, for instance, if they are exposed or buried, and then so we can study the local backbone dynamics using continuous wave or CWEPR spectroscopy, or if your protein is basically interacting with the membrane, we can determine the membrane immersion depth. But the main technique that we use in the lab is double electron-electron resonance or DR spectroscopy, which basically we introduce two labels and we can measure the distances between them using dipolar coupling, which inversely related to the cube of distance, so we can measure the distance between them. And the primary data that is obtained here has two components. One is the random intermolecular interactions that shows itself as an exponential decay. And by dividing this primary data to this exponential decay, we get this oscillatory function corresponding to the specific intramolecular interaction of interest. And then by fitting the primary data or this background corrected form, we can obtain distance distribution. And in different systems, when we have changes between the states A and B, distance changes could be a change in average distance. For instance, when the free energy between the two states, the difference between free energy is large, or in most of the biological systems, we have ensembles of the states at equilibrium, and equilibrium between the state A and the state B can be shifted by changing the experimental condition. Therefore, the distance distribution represents a snapshot of energy landscape of protein under a specific set of experimental conditions, and also we can determine change in the free energy of sampled state from the relative population. So that's also something that we could do using here, and this is our pulse EPR facility at St. Louis University, so we're lucky that we have that and we can perform all these experiments. So going back to the main topic of this presentation, in humans, SPNS family has 3 members, and SPNS2 serves as the major S1P transporter. And as I explained, majority of its function is extracellular. So SPNS2 is a potential drug target for modulating the entire S1P export and signaling. And in fact, in 2017, there was a large study published in Nature that they knocked out 800 different genes in mice to check for the effect on the metastatic colonization in lung. And SPNS2 was one of the most effective one in reducing the colonization in lung by inhibiting the egress of lymphocyte immune cells to those sites. So, and therefore, that suggests that SPNS2 could be an effective target for reducing the metastasis. These transporters belong to the major facilitator superfamily of secondary active transporters. They have 12 transmembrane helices organized in N-terminal domain and C-terminal domain, and each of these domains has 3 helix repeats that are involved by gene duplications. The first helices are called cavity helices. They come together and form the substrate binding cavity. The second helices are gating helices, and they form helix 2 and 11 and 5 and 8. They form the lateral gates that provides the pathway to the binding site. And the last helices are support helices, which they mediate the interactions with the lipids. So, and throughout my presentation, I go back to these categories at different points. So these transporters function by alternating axis mechanism of transport that was proposed by Jardetsky, and it's mentioned that using conformational changes, substrate binding site alternately faces different side of the membrane, either side of the membrane. And the simplest model of alternating axis for MFS transporter is rocker switch model, which as it's appear from the name, assumes that the N-terminal and C-terminal domain preserves their rigid bodies, and depending on the directionality of the transport for the ions and substrate, they could function either as symporters or as antiporters. And as you could see, rocker switch model only predicts presence of the outward or inward facing conformation, because we cannot have the occluded conformation that we know a lot of these transporters, we could have these intermediates, including the Spns family that we want to study. Here I'm showing the multiple.
Sorry,
Yes.
why can't they have occluded state?
Oh, because if they assume only rigid conformation, the N-terminal and C-terminal domain, the rocker switch says that they could just rock, right? So
But somehow they should have the alternating access mechanism, meaning that at some point you can't be open to both sides, right?
Yeah.
So well, how does it work? It's
So
open to one side,
yeah,
and then later to the other side, but then somehow there's
so maybe it's about a stable occluded state.
It's like a
or-
transition state that
Yes.
you go too fast.
Yeah, or you know, we know that, for instance, in the case of let's say Spns family, we really don't have for some of these members the obvious outward facing conformation. So
Okay.
it's non-canonical. Yeah.
Mhm.
So here I'm showing the multiple sequence alignment between three of the bacterial homolog and the Spns proteins from higher organisms. As you could see, the N-terminal domain on the left, we have a lot of conserved residues between both the bacterial homologs and the human and higher organisms, which implies that you know we could see some similarities in their transport activity. But for instance, the C-terminal domain is highly divergent between, especially the bacterial homologs and the human transporters or higher organisms. It's highly conserved between the higher organisms, but not for the bacterial homolog, which shows that there are also differences in their mechanism. In fact, some of them function as symporters, some of them function as antiporters. For instance, Spns1, which is located in the lysosomal membrane, is a proton lysophosphatidyl lysophosphatidyletanolamine symporter, which basically uses the pH gradient across lysosomal membrane, and it's a symporter. And Spns3, which we think that it's a multi-drug antiporter, so either we still don't know, but it seems that it's a multi-drug antiporter. HnSpns is a bacterial homolog from Hyphomonas neptunium that at the time that we started the project was the only one that is crystal structure available. And we assume that it's an antiporter, but now we have some preliminary transport assays that shows it might also function as symporter similar to the Spns1. So that's something.
What do they do in bacteria, by the way?
It's a marine bacteria.
Okay.
We have no clue.
idea. Okay.
And then I will come back to that.
I see.
Yes.
Is there some knowledge about that E. coli homolog?
About the DGOT or the
Yeah,
MDR from Mycobacterium smegmatis.
yeah. Do they
DGOT?
also transport lipids?
No, it's a degalactonate. It's a proton degalactonate. Actually, Bob Strott, they have a PLOS biology paper on that. And also there is another recent JBC paper that they solve the structures. And yeah, it's a symporter. It's an electrogenic symporter. So yeah, and the other one, as I mentioned, DGOT is a E. coli electrogenic proton degalactonate symporter. And the other bacterial homolog, which is the subject of our recent study that Ali and Ahmad we are collaborating on, it's a drug transporter from Mycobacterium smegmatis, and of course it has to be an antiporter to function as a drug transporter, as well as the human protein. So we started our study on this bacterial homolog from Hyphomonas neptunium, and as you can see between the human Spns2, especially the N-terminal domain, is highly conserved between the two, so we can basically have a mechanistic comparison between the two systems. Here I'm showing the crystal structure of the bacterial homolog. For instance, some of the important residues, we have this arginine 42 in the middle of TM1 that is a putative substrate binding site, so it can bind, let's say, lipids. And we have this glutamate 129 in the middle of TM4 that it was hypothesized to be the master proton switch to drive the conformational transition. And as you see the proximity of the two, it was hypothesized that the coupling mechanism is that when the glutamate 129 is deprotonated, it can form a salt bridge and interact with arginine 42. So arginine 42 is not released to bind the putative substrate, but when it's protonated, then arginine 42 is released and it can bind. As well as, protonation of this was hypothesized to generate the conformational transition. But in our study, actually, we could find that this aspartate 41 right before this arginine 42 could also function as a protonation switch, and we have other residues. On the right, I'm showing actually from in the last summer, at least 8 different structures of the human SPNS2 came out. So first, it was the AlphaFold model, and then the structures, when the structures came out, the first thing I did was I took the AlphaFold model and put the cryo model, 0.4 angstrom actually. And then I read the paper, what they have done. They took the AlphaFold model, put it on the map, and that was the basis for the entire solving the entire 5-6 different structures. So for the first time, AlphaFold, actually the cryo didn't add much. And it was funny that for cryo we cannot solve also the structure of the loops. So at least AlphaFold has the loops. So yeah. And as you could see also from these structures, that a lot of those conserved residues, especially the N-terminal domain of the bacterial homologs, are also conserved in the human one. So we could have a direct mechanistic comparison between the two systems. But at the same time, for instance, if we look at the inward closed conformation or occluded conformation, we could see that we have 2 sets of salt bridges that could form to stabilize the inward closed conformation, which in the case of bacterial homologs, interesting that we could have only 1 set of these salt bridges, and it's non-existent, and it's only appear in the case of the SPNS proteins from higher organisms. Another interesting difference between them is that in the case of the eukaryotic SPNS protein, we have this glutamate in the middle of TM10 corresponds to this glutamate in the middle of TM4, because if they are evolved by gene duplication, the corresponding transmembrane helixes tend to the 4. And you have at the same exact spot, we have that in the middle of TM10. And it's interesting to know what is the function of that, if that could also function as protonation switch. So it's a lot that we still can do using DR spectroscopy as well as molecular dynamic simulations to basically answer these questions. Another interesting example from the cryo structure, these are the 2 inward-facing cryo structures from 2 different labs. And sphingosine 1 phosphate is bound to its binding site. The nice thing is that the tail binds to the C-terminal domain and the exact same spot, and this is done by 2 different labs. But the head group in one structure faces toward the lateral gate between 5 and 8, so it seems that gate 5 and 8 provides a passage, either for the release or the entry of the sphingosine 1 phosphate. In the other case, the head group actually rotates towards those residue, that arginine that we talked, that although it's not interacting, but basically it's provide a pathway for the entry and interaction with the substrate binding site. And I will come back actually to this gate between 5 and 8. It seems that in different members of this family, this gate is important, either for the release or the entry of the substrate.
So you mentioned arginine 40 something in the beginning.
That's the arginine 119 in the human protein. The same arginine. The exact same arginine.
Because there was another arginine that was also highly, even more conserved.
Yes. You are
122
exactly right.
or something.
Arginine
122.
122. Arginine 122 is a part of the B-like motif, and basically by changing the positive electrostatic potential in that region, it could promote protonation and deprotonation of glutamate 129. And actually our MD simulations nicely showed that the arginine, how that
I see,
arginine could
I
affect.
see, I
Yeah.
see. Cool. Mhm. Okay,
Some of my slides, the figures are big, so.
yeah, it takes time to load it.
Okay, all right. So, for instance, here I'm showing our tier studies of the bacterial homolog from Hyphomonas neptunium. As I mentioned, to do the traditional way of tier, you have to remove the endogenous cysteine and hope that the cistless construct and the structural integrity of the protein is not affected, and we can use different methods to examine that, like CD spectroscopy, thermal stability, actually, tryptophan quenching, and functional assays. Those are very important actually to show that that's true. And then we can reconstitute the protein either in nanodisk of different lipid compositions. In this case, we use the membrane mimetic, plasma membrane mimetic composition without cholesterol. But the funny thing is that Hyphomonas neptunium doesn't have phospholipids. It has only glycolipids.
Oh.
And what we did, we tried to use glycolipids from chloroplasts, because those are the ancient ones. We couldn't prepare the nanodisk. So this is the closest one that we said, okay, we want to compare this to the human one. Let's put both of them in similar, but we removed the cholesterol because definitely they don't have cholesterol.
Can I ask if
Yeah.
this is the same composition you do functional assays in?
Yes.
Okay.
Yeah.
But it's not a nanodisk, of course. For the functional assay, when you want to make sure your protein is still functional after you remove all the cysteines. So what lipid composition do you use there?
Those functional assays, in this case, we use the bacterial system. We express those mutants in E. coli, and then, we
express it in E. coli.
Yes,
Okay,
yes,
okay,
yes,
gotcha.
yes.
Okay.
But you're right, actually, for in vitro transport assays,
Sure.
Yeah, but the protein was stable, so that's actually why we went with this. And then we reintroduced labels on the intracellular side as well as preplasmic size as double labels, and then we can measure distances between them under different conditions. So motif A is the most conserved sequence motif in MFS transporters. It's located between intracellular group between TM2 and TM3, and we have some residues like aspartate here or this arginine at the beginning of TM3. That these residues, in addition to other charged residues like this aspartate at the beginning of TM4, as well as the transmembrane helix 11 from the C-terminal domain, they come together and they form a structural motif A. And it's hypothesized that this structural motif A stabilizes the inward closed conformation of the protein. And there is also this charge relay network between this acidic, this charge residues between TM2, TM3, and TM4. That formation of this charge relay network also is important for the stabilization of inward facing conformation. Here, we start with those distance curves between the gates, intracellular gates between 2 and 11 and 5 and 8. And what we could see is that when you measure distances between them under basic pH conditions, the distributions that we get corresponds to the inward open crystal structure that was available. And interestingly, when we lower the pH to pH 4, so we have protonation of those acidic residues. In the case of gate 2 and 11, we have a change to the short distances corresponding of the formation of that structural motif A that we were talking about it. So we have closing of the intracellular side of the protein. But at the same time, the gate 5 and 8 cannot close completely. And by working on the other bacterial homolog, we could see that this could be one of the signatures of this family of the protein. Basically, the inward closed conformation, the gate 2 and 11 completely closes, but so it's asymmetric. Basic gating is asymmetric, and gate 5 and 8 actually stays open. as I mentioned, those even cryo structures shows that provide a passage for the lipids to get or the
But does it go to the inward? This is the inward side. You're looking at the cytoplasmic side now.
Yes.
So it closes enough for the substrate not to come back, right?
So it closes, for instance, in nanodist, we have distances around 25 to 30. And when we refined the model, the metadynamic model, the occluded model that Ali and Seper did, and we looked for tunnels and everything, 5 and 8 is still closed. Although, you know, it's not completely closed. It's occluded. So, but if we go actually to those distance pairs between the cavity helices, those helices that are involved in substrate binding, we could see that a structural rearrangement to longer distances. And when we did actually the modeling using MD and as well as other models, we could see that in addition, we have the closing of the intracellular side, but we have also the rotation and tilting of the cavity helices that correspond to this, longer distances. But overall, globally, we have closing of the intracellular side of the protein. So, okay, let's look at the same gates on the preplasmic side of the transporter, gate 5 and 8 and 2 and 11 on the preplasmic side. Again, on the preplasmic side, at basic pH values, you have distances corresponding to the outward closed conformation. And when we lower the pH, suddenly we see that the preplasmic sides become the gates, at least the gates become highly dynamic. So both the gates 2 and 11 and 5 and 8, and 5 and 8 is dynamic actually, all the way. And when we look at the preplasmic segment of this gating helices like 5, 8, and 11, we see that they consist of a lot of helix breaking residues that they could provide us with this flexibility. So, and then the nice thing is that this flexibility between the gates on the preplasmic side can provide passage, lateral passage for the substrate either to be released or reaches the binding side. So that's something that can do. But at the same time, when we look, there is a salt bridge between TM2 and 11 and 7 on the preplasmic side of the protein, which basically, as long as the salt bridge is in place, we do not have an obvious opening of the outward facing. And indeed, when we put labels before and after the salt bridge and measure distances between them or between those and other sides of the protein upon under acidic condition, we cannot detect conformational change, significant conformational change that we were expecting. So basically, the salt bridge stays closed, keeps the transporter in the outward closed, but at the same time, the lateral gates are highly flexible that provide a passage for the substrate. That's why we claim that it's a non-canonical alternating axis. It's not only obvious inward facing and outward facing conformation. We thought that maybe the lipid composition is the cause, because for a lot of this transporter, lipid composition can affect the stabilization of, let's say, outward facing conformation. And the only lipid composition alternative one was E. coli paralipid, but again, we couldn't detect any conformational change. And we concluded that the protonated state of the transporter is an occluded conformation, which at the same time, the lateral gates, the flexibility of the lateral gates provide a passage for release or entry of the substrate. And the nice thing is that the salt bridge is highly conserved between both the bacterial homologs and the human transporters. So the salt bridge is highly conserved between different members of this family. And then Ali and Seper, in order to have a better understanding of the coupling mechanism in this transporter, they took the inward facing conformation of this protein and they basically simulated different protonated state of those two acidic residues in the middle of the membrane. And they probed the salt bridge formation between this aspartate 41 and arginine 122, and the other one was between the putative protonation switch and the putative substrate binding site. So we could see that under these two conditions that aspartate 41 remains protonated. First of all, we have disengagement of the arginine 122, so it's released. But under these conditions that aspartate 41 is protonated, protonation and deprotonation of glutamate 129 cannot generate the type of conformational changes or disengagement of the arginine binding site that we were expecting. Also, the purpose of doing the MD simulation, especially the time scale, was also to probe local conformational changes, which can be complemented by global conformational changes that DIR can provide. So, and it worked actually very nicely in this case. For instance, what we could see is that as long as aspartate 41 is protonated, we can also not detect conformational rearrangement, local conformational rearrangement in the substrate binding cavity helices 1 and 4. And the other thing is that we don't see is the formation of that charge relay network that we mentioned, formation of that charge relay network between TM2, 3, and 4 is prerequisite for the stabilization of inward-facing conformation. The outward-facing conformation, inward-closed conformation. So, and the prediction is that if that's the case, we should not detect closing of the intracellular side as long as this aspartate 41 is protonated. And the nice thing was that we did MD simulations before the experiments. So, so yeah, it was really nice. So we experimentalists, we are always skeptical, but in this case, MD really beat us, and we learned a lot from those MD simulations. Whenever actually aspartate 41 is deprotonated, then it can form that salt bridge, which is stable, and upon protonation of glutamate 129, in fact, we could see that huge disengagement of that arginine, and then which then can bind the substrate. Also, the conformational changes, if you remember, for the cavity binding helices, substrate binding helices, it predicted that we have the tilt and rotation of those helices, and we could easily see that under these conditions, and the charge relay network between 2, 3, and 4 can form in both cases. So we predict that we have the closing of the.
So can I ask you a question?
Yeah.
How do you distinguish between a large tilt and rotation at the same time?
Oh, in this case, I just looked and I saw that, for instance, TM1, we have the rotation. How we can experimentally?
Yeah, experimentally.
So what we do, if we are lucky, one of our those spin labels that we put on the protein, upon rotation, it basically clashes or become less mobile.
Okay.
And we could exactly see that. So yeah. So we could see the rotation of those helices as well.
Okay. I see.
And therefore, so what we could conclude is that protonation of aspartate 41 regulates the intracellular master switch glutamate 129, and all of these local conformational changes that we predicted using MD happens while that salt bridge, preplasmic salt bridge, is intact. So that was another important conclusion that we had, that all of these changes in MD simulations happen while that preplasmic salt bridge stays intact. Now we go to the experiment. So what we have done, we basically use this protonation mimetic mutation of glutamate to glutamine or aspartate to asparagine to see if those observations that was predicted by MD, we could also detect it globally. And we could see that, for instance, for this distance pair between TM2 and 11 on the intracellular side that detects the formation of the structural motif and closing, when we mutate the glutamate 129, in fact, we see a shift to this short distance corresponding to the closing on different, independent of the pH value, which shows that really, this glutamate 129 act as a master switch for the conformational changes. In fact, when we have the D41N, we don't see that. So that mutation basically inhibits the closing, which MD predicted that actually it's happening. And when we have both mutation, we see that the effect of D41N is dominant than the E121Q on the closing of the intracellular side. Another mutation that we have done is D142N, which is the aspartate at the end of TM4. If you remember, that's a part of that structural motif A that form. And again, when that one is protonated or we have the mutation, we don't see the closing of the intracellular side. In the middle, I'm showing one of the pairs on the substrate binding cavity helices, 1 and 10. And again, when we have the E129Q mutations, we have a shift to the longer distances, which corresponds to the shift that we see independent of the pH. And then when we have the other one, we have a stabilization of the pH 9 state. So these two protonation sites, they have the opposite effect. So one is shifting the closing, one is stabilizing the opening of the protein on the intercellular side of the protein. And it was true also for the rest of the sites. On the preplasmic sites, we introduce the same mutations, but we cannot detect the same conformational changes so meaning that there's a big coupling of proton-dependent conformational changes between the intracellular side and the plasmic side. And at least those protonation switches cannot basically control the conformational changes on the or weakly control the conformational changes on the preplasmic side. So that's a distinct actual difference between intracellular side, that the conformational changes are basically controlled by the protonation and deprotonation of switches that we have, but on the preplasmic side, actually, we cannot see that. So we reconstituted, to test that, we reconstituted the protein in liposome. And remember, in nanodisk, we don't have the pH gradient, but in liposome, we could have pH gradient. And these were inside-out proteins. So if the pH inside was 5 and outside was 5 and 8, again, we cannot detect conformational changes, meaning that in the preplasmic side is weakly coupled to, so this proves that actually. So I don't say for the first time, of course, for many times that the MD simulations nicely corresponded to the experiments, but in this case, MD simulations actually taught a lot about the energy coupling between the substrate transport and protonation. So and we have a nice actually correlation between the experiments and the MD. Another nice experiments that we can do using DR spectroscopy is that by taking those distance pairs that are pH dependent, by plotting the population of rising or going down as a function of pH, we can determine the pK of conformational changes of the protein that reflects the protonation of those acidic residues. And we could see that for the intracellular side, we have a pK of 7.6, but for preplasmic side, we have a pK of 4.8. And we think that in the case of HnSpns, it functions as a symporter, and it's actually it's a homolog for the SPNS1, which is in the lysosomal membrane. What's the pH of inside lysosome? 4.8. Yeah, so it nicely actually fits the physiological function of these proteins. And in order to nail that, in fact, these two acidic residues are corresponding for that, we just did the mutation and removed the two or one of them, and then we see that the conformational changes completely abolishes on the intracellular side of the protein. And on the preplasmic side, these mutations, the same double mutations, cannot actually generate the conformational changes, which prove that we have this conformation. Okay. So Ali and Seper, they did the non-equilibrium driven MD simulations starting from the inward facing crystal structures. They stabilize and occluded conformation of the protein, and nicely the plasmic salt bridge, as well as the salt bridge on the intracellular side between TM11 and TM4, which was a part of that structural motif A, stabilizes the occluded conformation. They did the equilibrium simulations, and in fact, the protons could reach those sites using the main vestibule, as well as be transported to the intracellular side of the protein. And we can use dear distances to basically refine those models. So they took, for instance, either the inward-facing crystal structure or the occluded model that was generated using non-equilibrium MD simulations. And as you could see, in the case of crystal structures, we have a discrepancy between the dear, experimental dear, and what we have. But in the case of the occluded model, because it was occluded, it's a good actual correlation without even refining the
So unrefined is the result of the non-equilibrium driven.
yes.
Without
And
okay.
it was equilibrated shortly after the
Without
just
the data.
dear,
I see.
And
I
then
see.
when we use it, use REMD, actually restaurant ensemble, MD simulation, now we have a nice fit, and we could show that the X-ray structure inward facing correspond well with the refined inward facing model. And in fact, we have an occluded actually conformation. And as you could see, although here I wanted to emphasize that still the gate 5 and 8 is not closing completely, but the distances are very, they're not. So in fact, it's an occluded conformation. So we might have other passages that we propose in our publication that for the proton transport, but yeah, that's something that down the line probably we'll get to them and study them more carefully. So yeah, so it seems that basically the gate 2 and 11 completely closes, but gate 5 and 8 does not, and which in the case of the second bacterial homolog, which I hope actually I will get some time to present some of those data. All right, so we skipped the substrate, right? One of the reasons was that we don't know what is the substrate of this bacterial homolog, but we could see that a lot of those residues in the binding cavity are conserved between them. So we don't know the substrate for HnSpns, but we know the substrate for SPNS2. And the first thing what we did was adding S1P, sphingosine phosphate, to the bacterial homolog. We couldn't detect any conformational changes. And then the second bacterial homolog in our study, this drug transporter from Mycobacterium smegmatis, it can confer resistance to capreomycin, which is a tuberculosis drug, as well as a bit to ethidium bromide, but it's susceptible to rifampicin, another important antibiotic against tuberculosis, and said, okay, let's see that. And then because we wanted to study the MsSpns, the second bacterial homolog, Sam, my postdoc in the lab, basically used E. coli, express the protein, and we could see that it can indeed induce resistance against capreomycin and ethidium bromide to a lower extent, but not for the rifampicin. And then what we did, we took this pair, intracellular pair between TM1 and 10, which is the substrate binding cavity. So it should be sensitive to the substrate binding. And we see that for all those compounds that are putative substrate, you cannot detect conformational changes. For rifampicin, basically, it can stabilize the inward facing conformation of the protein. And when we actually tried it with verapamil and Hoxt, because I study molecular transporters, so I just put all those lipophilic compounds, in fact, actually stabilize the inward facing conformation, which when we go from pH 7.5 to 9, the effect is much higher. And only when we have the mutations or protonations, we can detect closing of the intracellular site. So substrate binding, it seems that cannot generate the conformational changes, and only proton binding and protonations is the main driving force. But this is nice because one of the cryo-EM structures of the human transporter showed that inhibitors basically stabilize the inward facing conformation. So, and using DEER, basically we can screen a large set of inhibitors or those important ones to see actually which ones are effective modulators. And we are excited that in the future, actually, we could use that for in our advantage. So yeah, what we see to just summarize this part of the presentation. So what we see is that upon protonation, we have flexible preplasmic side of the protein, and we have closing of the intracellular side of the protein. And what will happen is that upon aspartate 41 protonation, it promotes the deprotonation of glutamate 129 because arginine 122 is released, so we have positive electrostatic potential that results in deprotonation. And upon deprotonation of the glutamate 129, the occluded conformation is destabilized. So we have the transport of the proton to those acidic residues in the structural motif A. So we have opening to the intracellular side. And we have a non-canonical alternating axis. We have only occluded as well as inward facing conformation of the protein. And then what will happen is that the proton transfers to this glutamate 129, which then releases the arginine for the substrate to come and bind to it. And upon protonation of glutamate 129, again, we have the conformational change. And remember, in our paper, actually, we didn't have any transport assay. So we proposed this model as an antiport model. If you take the same proton transfer pathway and then add the substrate from the extracellular side that could reach the binding site through the gates, flexible gates between 5 and 8, especially the lipophilic compounds that they could go to the membrane, we could have a symporter actually, that proton transfer. So this model is also consistent for the symport mechanism between different proteins. We have the HnSpns now. If I have a couple of minutes, I'm sorry that I'm talking with.
That's okay. We're just gonna leave some couple of minutes for questions.
Sure. So yeah, what you see, for instance, in the case of HnSpns, the first published study is that under basic conditions, preplasmic side is structured and the cytoplasmic side is more flexible. In the case of MsSpns, the other actually bacterial homolog is flexible on both sides. When it's deprotonated, it's highly flexible. Upon protonation, in this case, we had actually a higher flexible preplasmic side, but intracellular side was not. But in the case of the bacterial homolog, we have a shift to the shorter distances, meaning the closing of the overall closing of the preplasmic side, but we have a shift to the longer distances on the intracellular side, which makes sense. If it's a flux transporter, it has to close upon protonation, it has to close and open up for the substrate to reach. But the main actually difference and similarities between the gates 2 and 11 and 5 and 8 for both of them. In the case of TM2 and 11 on the HnSpns, we have complete closing of the or we have closing of the preplasmic and intracellular gates. But in the case of the MsSpns, we don't have complete closing of the gate upon protonation. And we were wondering actually why that's the case actually. And here actually what I have done, the shaded area is the AlphaFold model of the MsSpns that was the only available one. And so if you compare that, yeah, you see that we don't have complete closing of the gate intracellular or preplasmic gate between 2 and 11. In the case of 5 and 8, actually it's interesting. We have an opening of the gate. So under base acidic condition, we have the opening on both sides, especially the intracellular side. So gate opens. So that's something that it's worth study. So intracellular gate 2 and 11, we introduced the same protonation mimetic mutation of the glutamate 129, which is 126 in this case. And in fact, as you could see, we have a slight shift to the shorter distances. We have the closing of the gate. So this glutamate is supposed to be the master protonation switch. And it's interesting because, as you could see in the case of MsSpns, this aspartate at the end of TM4 is mutated to arginine. And when you look at the corresponding residue that it might interact, it's a phenylalanine. So basically, it's a salt bridge is replaced with a cation pi interaction. And because this is an arginine, so there's also another arginine that we know that it comes from TM11 and interacts with this. Maybe we also have an arginine-arginine pairing. So it's nice that, you know, the nature of the non-covalent interaction changed, but you know, nevertheless, we have the closing of it. And that's something actually unique that you could see that. If you remember, in the case of HnSpns, we have the opposite effect of protonation of D41 and E129Q. The same is true in the case of the MsSpns. E126Q closes the gate 2 and 11. D38N keeps it in the open conformation. It's a slight change, but that corresponds to opening and closing of those gates. And then when we have the double E2Q mutations, it still actually stays open. So for opening and closing of this gate, D38N is the dominant one. And here I'm just representing this result differently based on pH and then mutations. And as you could see, again, E126Q closes the gate 2 and 11, but D38 and actually keeps it open. So that's something that it's similar between HnSpns and MsSpns. The gate between 5 and 8, D38N closes the gate, but E126Q opens the gate. So if we look at the data, then we see that upon protonation of E126Q, we have closing of the gate 2 and 11 and opening of the gate 5 and 8. In HnSpns, we had the closing of the gates 2 and 11. The 5 and 8 didn't actually close completely, which means that actually it might be a signature of this family of transporters. So it's very nice when you could do mechanistic comparison between different members. So you could see similarities. And then the PK of conformational change for the wild type protein is 8. So, and we think that it might actually reflect the protonation of D38N. If you look at some pairs between the TM4 and 11 on the intracellular side, I think I should stop actually shortly. So the binding actually cavity, again, what we could see that the binding cavity, E38N closes the binding cavity, but E126Q opens it. And in this case, in the case of the binding cavity, E126Q, it seems that it's a dominant one, because when we have the double mutation, that one. So it's very interesting. And the PKs that we say is PK for the wild type is 6.9, but for the E126Q and E38N goes down to 6. Yeah. So it's completely interesting, something that we could see. And on the preplasmic side, I promise I have only 2 more slides. So sorry that, you know, when I'm excited about our results, I have to present it. So all right, on the preplasmic side, for HnSpns, we cannot detect conformational changes. We had that recoupling that we mentioned, but in the case of MsSpns, it's strongly coupled. So basically, E126Q closes the preplasmic side between these are between 1 and 11, the gate, and D38N opens it up, and D38N is the dominant one between them. Do you remember that salt bridge? So one of our reviewers was upset that we didn't mutate one of those residues to stabilize the outward open conformation. And we told them that as soon as we touch those, we cannot express the protein. So in this case, actually, we went to 36 liter of culture just to get some to do the experiments. And what we could see in the case of the MsSpns, indeed, as soon as we have this E57N, we have the shift to the longer distances on the preplasmic side and the intracellular side closes. So in fact, that salt bridge keeps the protein actually in the outward closed conformation. And all of those conformational changes that I mentioned here happens in the presence of that salt bridge. Because the distance changes are between 20 and 30, but as soon as we disrupt that salt bridge, the changes go to let's say 40 and 50. So all of those conformational changes involved in the transport are happening for that. My last slide, the first results that we have on the human transporters. So after all 8 cryo-EM structures, we were upset that yeah, they have done a lot. So what we did, basically we put spin labels on TM1 and 10, the substrate binding cavity, and what we could see for the apo pH 9 state, the distances that we see correspond to the inward open conformation. Although another thing is that actually the protein is highly dynamic, right? It samples a large conformational space, so that's one thing. So we lower the pH to pH 7.5. Again, no changes. We added the sphingosine phosphate, which this inward facing conformation was all in the presence of the sphingosine phosphate. We cannot detect conformational change. We lowered the pH, and we could see actually the shift corresponding to one of the cryo-EM structures, which is the inward flows are occluded conformation, meaning that the protonation is the driving force for conformational change. The Cell paper that was published says, okay, it's not proton dependent and it's facilitated diffusion, but even one single deer experiment could show that actually, yeah, indeed, and that's the beginning of the next 1 or 2 years that yeah,
it's 5 years.
Absolutely. All right, thank you so much. I'm so sorry that I spoke. Yeah,
No, that's it.
thank you.
Okay. Thank you very much. You obviously love this protein and you know too much about this protein, but that was a very good example actually how sensitive biophysical measurements like EPR and NMR match very nicely with MD, but very good example of how we can learn from each other. So I had a couple of questions myself, but anybody else can start while I look them up. Or maybe I can actually start with glycolipids. Do you know what glycolipids do you have in that
HnSpns? We have some guesses, but since we need to synthesize them, what I'm thinking about to go back, take that chloroplast once, even if we cannot generate the nanodisk, maybe I have to contact Dr. Slager actually to see. So because as soon as we add the biobits are supposed to remove detergents, right? It seems that they are also removing the glycolipids or something. So what we can do, we can purify the detergents and add the lipids, the glycolipids from chloroplast and see. Yeah, that's a very interesting.
I was wondering what do you expect from glycolipids? What functional effects might you expect? Do you expect anything in glycolipids?
It's hard to talk about it. Yeah. For instance, in the case of the human SPNS2, we know that phosphonosides are like POPI, PIP2, they basically stabilize certain conformations. There's a nice paper that we discussed about it. And we have POPI in our compositions, for instance, in the case of that bacterial homolog. But it's interesting if we can basically get the native lipid.
I see. Because I mean, I can tell you the story over dinner. So any other questions? You guys,
Yeah, it's also related to glycolipid. I'm not sure about the membrane composition of that Mycobacteria.
Smegmatis?
Yeah,
We use E. coli for our lipids. Oh, no, I didn't mention. We use E. coli for our lipids.
Right.
For the second, because we know that it's from Mycobacterium, and usually, traditionally, people use E. coli for our lipids
Right.
for.
so I have a speculation that, because I'm not familiar with that particular Mycobacteria, but I heard of, for example, some other Mycobacteria, membrane are very rich in this, phosphoinositol monocytes.
Oh, right.
Yeah, and yeah, that's another kind of
Sure.
negative charge, glycolipid, very.
I hope actually Alan T has it available and or,
Yeah.
yeah, or if you know any sentence, yeah.
Computationally, we can do it.
Combination, yeah, of
That's
course.
why we are asking.
So then my speculation is that you see more disorder of the protein
Sure.
compared to the
Sure.
other bacterial homolog. Might be, E. coli lipids for that,
Sure.
protein is too soft.
Okay, for the second one, for
I
the,
see.
Right,
Yeah.
and that's,
yeah.
So I noticed you have a structure for the second one, right?
For
There
the
was a
second one, we have
PDB
only the
Yeah, this
alpha
one. They go
the
back.
alpha model.
Oh, no, there was a PDB code. If you go forward,
Oh,
the one, if you, one more,
and one more.
this one, what is
Oh,
this?
that's
human.
that's the human protein.
That's it. Oh, for the human protein, you have a PDB.
Oh, yeah, all those
Oh,
H
on the
structures
H
are, yeah.
That's great.
Oh, yeah.
So, by the way,
exactly.
so, you're done with your question, so this thing is a multi-drug transporter, according to your hypothesis.
And the SPNS3,
SNS3,
yeah, we hypothesize is
okay,
the.
but then that means non-specifically acting on multiple substrate, probably transporting other lipids too,
Sure.
beyond the spindle.
Yeah, and we see that, this gatings that we have on the extracellular side between 5 and 8, or intracellular side again between 5 and, these are lateral gating.
Yeah,
And in the case of, for instance, HnSpns, as soon as we add the lipophilic drugs,
Yeah.
we see conformational change and stabilization.
Right.
Yeah. In this case, also they added this potent inhibitor of human SPNS2, they call it 16D. It stabilizes the inward facing
Okay.
conformation of the,
Interesting.
so it could be actually,
Yeah.
and in this case also we bought all those inhibitors and drugs available against it, so GR could be a good method actually
I see.
to see. And the interesting results is to add them and lower the pH and see if they can inhibit the conformational
I see.
changes that we see. And as well as all those combination of protonation mimetic mutations, yeah.
Yeah, but again, I was going to comment on that.
Yeah.
When you use a protonation, yes, never mind.
Mimetic,
So you lose the salt bridge formation capability completely. So you might form a hydrogen bond at some point.
Sure.
But the negative charge is permanently gone when you go from aspartate to aspiration.
yes,
So
that's
you lose that. state,
absolutely.
essentially.
Yeah, but
Yeah, sure, but that's the way to approach the system.
Absolutely.
Any
That's
other
why
questions?
actually we do MD.
Yeah, exactly. We don't need mimetics, we just do it. Any questions on the Zoom? I know that quite a few people, oh,
I
Moin.
have a big picture question, I guess. First, I thank you for the talk and very sort of detailed investigation of this protein. My question is, how can identifying this like mechanism, pH dependent transport, or proton coupled transport, how can that inform therapies for going back to like metastatic cancer to either encourage or discourage transport
Sure.
with the knowledge of pH dependent?
Yeah, so we thought about it because the paper showed that they had this cell-based assay that they had the gradient, sodium gradient and proton gradient, and they couldn't see the simulation of the transport of sphingosine phosphate. That's why they concluded that it's a facilitated diffusion in both directions. But we think that if there is a pH dependent conformational changes and transport, it could happen under certain conditions like hypoxic cancer that we have acidosis, extracellular acidosis. So extracellular pH goes down as well as the inflammatory disorders. So in those cases, in inflammation, you also have extracellular acidosis, and that could actually regulate the function of the protein cells. Yeah.
So, okay. Good. So if there are no more questions, so thank you everyone. No, it wasn't too bad. If we skip 10 past 4. And you still have time, so